Let's get started with chapter 9. So, chapter 9 is all about lipids and biological membranes. And we are, actually, before we get started, a few reminders as usual. So, tonight, if you had to do a revision for your paraphrasing assignment, they're due by tonight. So, if you haven't done your revision yet and you need to, please do it. Everyone's quiz 6s are in, so quiz 7 is the next quiz that's due. That's due on Thursday, and it's about chapter 8, which we finished last Thursday, so that should be enough time for you to review everything and get that sent in. This week we're going to do chapter 9, and we're going to hopefully start and get through some of chapter 10, and we'll finish up next Monday chapter 10 and any exam review type questions you have, and your exam is next Thursday. So exciting. Yeah, we live in exam. So today we'll start with chapter 9, and we'll start with the questions on lipid classification. So let's just start by reviewing what is a lipid. What are some of the common characteristics of lipids? And anyone can just raise their hand and shout out some characteristics. Yeah. Non-polar. Yeah, excellent. Another word for that is hydrophobic. That's very illegible. Hydrophobic. All right. Yeah. Sure. Yes. No, I just changed up the point system, so it's more like your exam. So like questions can be worth multiple choice. All the quizzes are weighted the same though, so it's not like this quiz is weighted more than each quiz is weighted equally for your final quiz grade. It's just that there's a more fine gradation of points, and I'll do that for the quizzes going forward. So the grading for your quizzes is more like the grading for your exams. I feel like that's helpful for you to get a sense of like I should spend more time on a high value question or spend less time on a low value question. It's not but it's not gonna like count for more towards you. Like yeah. Yep. Okay, so yeah. So lipids are nonpolar or hydrophobic. So what makes them nonpolar and hydrophobic? What kind of atoms are they made of? Yeah. Yeah, exactly. They have high hydrocarbon content. And what does that mean in terms of what kind of environments they like to be in? Where are they soluble? Yeah. Yeah, so they're soluble in nonpolar and organic solvents. And there's one other interesting thing about lipids that makes them very different from some of the other types of molecules we've studied so far, like nucleotides or like sugars or proteins, amino acids. So it's hard for me to ask this question without giving it away. But so things like nucleotides and amino acids and even sugars, they can all be made into polymers, right? They can form covalent bonds among single units and form multiple units. Can lipids do that? No, they cannot. Any other things we should know about lipids? Yeah. Yeah, so they're an important components of the cell membrane. I wouldn't say that's a characteristic of lipids, that's more like a function of them. But yes, that is also true, and we will definitely go into that. It's okay if no one has anything else. I'm just wondering if anyone thought of anything else. All right, so let's now move on specifically to fatty acids. So first of all, what does a fatty acid look like? Yeah. Yeah, and what makes the polar end more polar? Yeah. So do you remember what kind of functional group that is? It is a carboxylic acid. Yeah, so we call them fatty acids, right? One end of it is a carboxylic acid. So for fatty acids, they have a lipid nature, right? Because they have a long hydrocarbon chain. But on one end, they have a carboxylic acid, which makes that one end a little bit more polar. But for the most part, the rest of the fatty acid is pretty nonpolar. So now let's start looking at this particular fatty acid, which is labeled alpha-linolenic acid. And everybody can take a couple of minutes and think about these different terms you might want to use to describe it, and pick out which term or terms out of this list can be used to describe alpha-linolenic acid. So everybody take a couple minutes to think about it, and we'll go over it together. Some people are done thinking, some people are still thinking. Does anybody want to tell me any one of these terms that we can use to describe alpha-linolenic acid? Yeah. Yes, it is polyunsaturated. And how do you know that? Yeah, exactly. So we have one over here, one over here, one over here. So more than one double. So saturated is no double bonds. Monounsaturated is one double bond. Polyunsaturated, multiple double bonds. All right, how else can we describe those double bonds with these lists? Yeah. Yeah, and all of them are cis unsaturated. And how do you know that? Yeah. Yeah, exactly. So they are polyunsaturated. All those bonds are cis unsaturated. And are there any other terms? Yeah. It is omega-3. Yeah. And how do you know that? Exactly. Yeah. So if you start counting from the end of the fatty acid or the omega end, and then you count up to the first double bond, that tells you your omega number. So this is carbon number 1, carbon number 2, carbon number 3. Carbon number 3 has a double bond. It's omega-3. Any questions about this so far? Cool. So now let's talk about fluidity in fatty acids, which has a lot to do with those hydrocarbon tails. So just to get everyone familiar with how this question is set up, so there's 6 different questions on each line, and for each line you should choose the option, which is out of either 2 or 3 options of what types of fatty acid would be more fluid. So everybody take a couple of minutes and look through those and decide for those 6 properties which would make a fatty acid more fluid. And we'll go over that in a couple of minutes altogether. Alright, so let's start with number 1. Would fatty acids with a higher or lower melting point be more fluid? Yeah. Yeah, and why is that? Yeah, basically. I mean, if you think of something that has a lower melting point, it's going to be liquid at a wider range of temperatures than the one with a higher melting point. Can I give it that way? The more times, the more likely it is to be a liquid, the more fluid it is, right? So how about number 2? Yeah. Yeah, it's polyunsaturated. And why would that be? Right. And so what is that? What is unsaturation due to the shape of the fatty acid tail? Anybody? Yeah. Yeah. Yeah, exactly. So on double bonds, particularly the cis-oriented double bonds, are going to introduce a pretty sharp bend or kink into the fatty acid tail. And this is going to make them just physically unable to pack as tightly as fatty acids that have more freedom of rotation, so the ones that are more saturated. So if you think of your saturated fatty acid, that is a really bad picture. It's mostly going to be able to, basically can be flat and you can pack them together fairly tightly. But if you have somewhere in there a double bond, that's going to introduce a bend or a kink that would help or not help this fatty acid be able to squeeze in as tightly with its neighbors. Not my greatest picture, but there were better pictures in the lecture. Okay, so number 3, shorter or longer ones are more fluid. Shorter, yeah. And why might that be? Yeah, essentially. So the longer ones have more opportunities to essentially pack in with each other and form more of a more tightly packed area compared to shorter ones, which are maybe more easy to separate because they have fewer interactions with one another. All right, so number 4, cis or trans unsaturated. Yeah. Yeah, exactly. And that's for the same reason we just sort of described. So if it's cis unsaturated, you might have a really big bend that makes the fatty acid sort of turn the other direction compared to a fatty acid that is in the trans configuration. It will resemble a saturated fatty acid more and be able to pack relatively more tightly than a cis. So, all right, so how about tightly packed versus loosely packed? Kind of gets into what we've been describing for all the rest of them. Yeah. Yeah, exactly. The more loosely packed it is, the more fluid they're able to be because they're not interacting as much with each other. So that brings us to the last one, animal sources or plant sources. Yeah. Yeah, and why would that be? Yeah, exactly. So animal sources tend to have more saturated, and a lot of fats that we see from animal sources are solids, things like butter, or in meat when you see fat deposits, they're solid. Compared to plant sources, they're more unsaturated. If you think of a lot of the oils we use in cooking, a lot of those are from plants: olive oil, canola oil, peanut oil. There are some exceptions like coconut oil is a solid, but for the most part, a lot of plant sources of oils are more liquidy than animal sources. Any questions about fluidity? All right. So now now that we've talked a lot about structure, we're gonna bring that into function. So everybody take a couple of minutes and consider each of these 4 functions and match all of the lipids to them. So out of 1 through 4, some of them might have just 1 letter, some of them might have more than 1 letter. Everyone has at least 1 letter. So go ahead and take a few minutes and match up those functions with the lipids. Okay, so let's start talking about these different functions. So let's start with number 1. So what lipid or lipids are used for energy storage in plants and animals? Yeah. So no, it's not glycerophospholipids. Those have a different functions. What's that? D. Yeah, triglycerides. Yep. So triglycerides are very relatively efficient for energy storage, and it has to do with their structure, which we will talk a little bit more about in the next set of questions. But yeah, triglycerides are able to pack together fairly tightly, especially when they're made of more saturated fatty acids. And so that it makes them very good at storing energy, because not only can they pack tightly together, they're able to exclude water, which makes them take up a lot less space, and just generally be very efficient at the space and the types of and also the types of molecules in them, right? So saturated fatty acids are mostly reduced, which means they have a lot of space for opportunities for oxidation, and that's how we get energy out of them, essentially, is by turning the hydrocarbons into carbon dioxide through a series of reactions, or oxygen. But anyway, we'll get more into that as we go. So let's do number 2. So which one of these, or more than one of these, is a major component of biological membranes? Yeah. Cholesterol is one of them, yep. Any other ones? Yeah. B, yeah, glycerophospholipids, yep. Is there another one? Yeah. It is not C. Steroid hormones don't tend to be found in membranes. They can pass through membranes, but they don't live there. Yeah. Sphingolipids, yeah. So sphingolipids and glycerophospholipids are a big component of the actual lipid bilayer, and then there's a lot of cholesterol in between, in the middle of the lipid bilayer, helping to maintain fluidity. So those are all very important components of biological membranes. All right, so how about number 3? Which ones of these are molecules that can disrupt membranes and lyse cells or act as signaling molecules? It's a big job. Yeah. Not glycerophospholipids, but glycerophospholipids are mostly structural. They're used as a structure. Yeah. Yeah. Yeah, lysophospholipids, which are essentially modified in some ways glycerophospholipids. They're not too far off. But yeah, so lysophospholipids are when you have a phospholipid and a phospholipase enzyme cleaves either one of the fatty acid tails or a part of the polar head. That's what makes it a lysophospholipid. Lyso meaning like it was cut. And so because they're missing sort of part of their structure, they're able to sometimes pass through the center of a cell membrane, which could disrupt the bilayer and potentially puncture the cell or disrupt the membrane. And they're also really important for some signaling pathways that if we don't get to in this class, you will almost certainly eventually learn in another class. All right, so let's talk about number 4. So which one of these just acts as a signaling molecule, specifically between different types of tissue? Yeah. Yeah, so here's where the steroid hormones come in, exactly. So steroid hormones are really great because they can pass right through cell membranes, but they don't live inside them. They're sent in between different types of cells and different types of tissues to essentially send messages to other places. All right, any questions about lipid functions? Great, cool. So let's now focus more on those structural components. So we got that same basic set of lipids, except now I'd like you to match up each lipid with all of its structural components. So take a few minutes and match everything up. so let's start with number 1, triglycerides. What are triglycerides made from? Yeah. Yeah, exactly. So they start out with a glycerol as their backbone, and then they have 3 fatty acids. So triglyceride, tri for 3. The glycer should remind you there's glycerol in there. Yeah, and then this brings us again back to why triglycerides are really good at storing energy. They have 3 fatty acid tails on one glycerol molecule. The fatty acid tails can be packed pretty closely together, especially when they're saturated. And in addition, if they're long hydrocarbon tails, they're very reduced. And when they pack tightly, they can exclude water from their storage area, which makes them very space efficient as well as energy efficient because of all those carbon-hydrogen bonds, which are highly reduced. Okay, so let's talk about glycerophospholipids. What do those look like? What are they made from? Yeah. Yeah, so glycerophospholipids also start with a glycerol, hence the glycero in the name. They have 2 fatty acids, and the 3rd fatty acid has been replaced by G, a phosphate-containing head group. All right, how about number 3? What's cholesterol made from? Yeah, E. Yeah, so cholesterol is essentially a fused ring structure made from 4 rings that are fused together. That is the steroid backbone, as we call it. So it makes cholesterol and also makes something else in this list. Yeah, so let's jump right into number 4. What does number 4 structure look like? Yeah, also E, excellent. So yeah, same 4-ring fused structure, makes cholesterol and steroid hormones. They have a lot in common structurally. All right, so that brings us to number 5, the sphingolipids. Possibly the most fun to say, and maybe the most fun structure if you read to them. So what are sphingolipids made from? Yeah. Yeah. What else are they made from? Yes, exactly. They have 1 fatty acid residue. So sphingosine by itself does not include that 1 fatty acid residue. Sphingolipids are sphingosine plus a fatty acid residue. And then there are a number of other sort of variations on this theme that we'll talk more about in the next slide when we're talking about all the different sphingolipids. But they all start off of this basic backbone, which is sphingosine plus fatty acid. That is a sphingolipid. So while we are here thinking about all these different types of lipids and what is present in our cell membranes, what particularly of these components do all membrane lipids definitely always have on them? Yeah. Yeah, so they don't always have glycerol, but they do always have that polar head group. And fatty acid tails. Sometimes I write fatty acid Fa. Hopefully that's not confusing. So that's sort of when you think about a lipid bilayer, right? You have the outside part, that's the polar part that likes water. So it always needs some kind of polar head group that's interfacing with water on each side. And then you need the middle part, the nonpolar part, that is always going to be made of some kind of fatty acid tail. And the specifics of what all those things are does vary, but that's every lipid component of a lipid bilayer is going to have those components to it. And this makes them amphipathic, right? They have some nonpolar part and some polar part. All right. Any questions about any of this before we start talking more about sphingolipids? All right, so let's talk about sphingolipids. So take a couple of minutes. Here are some of the 4 of the big sphingolipid categories that we talked about in the lecture, and some of the components they might have. So take a few minutes and match these all up. All right, so it looks like most people are done working. So let's start with, actually, does somebody want to tell me which one is the smallest one? And we'll start with that one. Got a 1 out of 4 chance of being correct. Yeah. Ceramide, yeah. Ceramide is the smallest one. So what are the components of ceramide? Yeah. So ceramide is just sphingosine plus fatty acid. And ceramide is a component of all of the other ones. So all the other ones are ceramide plus something else, right? So let's talk about what those something else's are. So let's start from the top. So what does a ganglioside have in addition to sphingosine and a fatty acid? Yeah. Yeah, it has an oligosaccharide. What about sphingomyelin? Yeah. Yep, it's got a phosphate-containing head group. And how about cerebroside? Yeah. Yeah, it's got a monosaccharide. So I know these names can be a lot or kind of confusing. One trick that might help you is the 2 that end in side both have sugars. Other than that, just gotta know what they're made of. Ceramide's the smallest one. And the other one's built from there. So just as a little review, let's look at all of these different options. So starting from the left, we have just sphingosine. And if we recall, sphingosine has this backbone that has an amino, it has 2 hydroxyl groups, and it has this trans double bond right here. So that's the sphingosine backbone, and it sort of starts you off with one kind of long hydrocarbon tail, but it is not a fatty acid, right? It doesn't have any carboxyl group that attached it. It's just part of the backbone. Ceramides have, in addition to sphingosine, have this fatty acid residue that's over here that's attached to the amino group. Sphingomyelins start with ceramide, but they have that phosphate-containing head group that we see here. Here's another type of sphingomyelin. So sphingomyelins tend to have either phosphocholine, which is the one I circled on the left. They also frequently have phosphoethanolamine as the other phosphate-containing head group. Those are 2 sphingomyelins. And then cerebroside, we're just changing out this head group. So instead of a phosphate, now we have 1 sugar for cerebroside, or a bunch of sugars for a ganglioside. Everybody feeling comfortable with the differences between all the sphingolipids? I know it's a lot, but yeah. Yeah. Yep. Yeah, I won't ask you to like draw one from memory, but yeah, for sure be able to, if I give you a picture, tell me what kind it is. Yeah. And you don't have to know the names of those 2 phosphate-containing head groups, but they'll come back for you later if you take more biology classes, I'm sure. So, yeah. Yeah. Good? Okay. Cool. Any other sphingolipid questions? Awesome. So that brings us to a picture where we're going to compare glycerophospholipids and sphingomyelins. So everybody take a few minutes to look at these 2 structures and tell me which one is a glycerophospholipid and which one is a sphingomyelin. And start thinking about all the different structural components that tell you which one's which. And yeah, I'll give you a couple minutes for that, and then we'll go over it together. Yeah, do you want to get us started? Yes, the bottom one is the sphingomyelin. That is correct. So here we have a yeah, so now we know for sure which one's which. So let's go into how we tell the difference. So do you want to point out one of the structural components? Mm-hmm. Yeah, that's a really excellent way to start. So glycerophospholipids have those two fatty acid tails that are each connected by this ester bond that we see. And the sphingomyelin only has one type of connection like this with a fatty acid tail. It's actually an amide bond, not an ester bond, right? So what else is different between them? Yeah, sorry. Mm-hmm. Yeah, you mean the one on the backbone of the sphingomyelin? Yeah. Yeah, exactly. So a sphingomyelin has this whole sphingosine backbone or sphingosine backbone, right? That consists of this hydrocarbon chain that has a trans double bond. And then the whole rest of the backbone also includes this kind of amino group and these two alcohols, right? There's a trans double bond. We have this amino. And that's very different from the backbone of our glycerophospholipid, right? What's the backbone of our glycerophospholipid? Yeah, it's glycerol. So glycerol is these three carbons here, and each one of them has or had a hydroxyl group on it. So the glycerol backbone has started with 3-OH, right? The sphingosine backbone had 2 original hydroxyl groups and 1 amino group. What else is left on these 2 different structures? Yeah. Yeah. Do you mean for the connection to the fatty acids, or do you mean to the other part? Okay. Yes. Yeah. Yeah. And that has to do with the fact that this one has 1 fatty acid and that the other one has 2 fatty acids. Yeah. Yeah. Is that what you meant? Okay, cool. Yeah. Is there any part that perhaps these structures have in common that we have not addressed yet? Yeah. Yeah, exactly. So they both have a very nice phospho-containing head group. And this one happens to be phosphocholine, which you do not have to remember the name for. There is something interesting about phosphocholine. It has one negative charge and one positive charge, giving the whole thing an overall neutral charge. What do we call that type of molecule? A zwitterion. Yeah. So which part, the left or the right of both of these, is the nonpolar part or the hydrophobic part? Yeah, the left. So this part, call this the hydrophobic region. And that would make the other side, the part on the left, the hydrophilic. Any other parts that we have not discussed yet? Or any other questions about any of these parts? Yeah.
So for the general structure of a phosphate with an alcohol, right? Is that true? Is that how it's supposed to be? Or
For the glycerophospholipid. Yeah, so the it's made of, the backbone is glycerol. Glycerol can attach to 3 different things. So the first 2 are fatty acids, and the third one is some kind of polar head group, which I believe always contains phosphate. Yeah.
so where does the alcohol like interface with like let's just say the choline? I was curious where the OH goes.
So the OH from the glycerol would be here, right? Here's 1, here's 2, here's 3. Can you see those? Right. So the first 2 are in traditional ester bonds with the fatty acid. The third one is in this phosphate ester bond with the phosphate group. Any other questions about these structures? See some thoughtful looks on people's faces. Hopefully that's a good sign. So let's keep going while you ponder all of these things. So we'll switch gears a little bit and we'll start talking about fat-soluble vitamins. So take a look at these 4 biological functions and match each one up to a vitamin. And then we'll go over it together in a few minutes. Yeah. All right. Looks like some people are still thinking. Some people are maybe done. But let's get started. So, so the first thing you should notice about this question is that it is asking about fat-soluble vitamins. So right away, there are 6 vitamin choices. We can immediately eliminate some of them because they are not fat-soluble vitamins. Does anybody tell me which 2 are in this list are the water-soluble vitamins? Yeah. C is one. B is the other one, yeah. So these are both water soluble vitamins. So they are definitely not the answer to the question about a fat-soluble vitamin. So now we have 4 vitamins left and 4 functions, so see if we can make this work out. So number 1, which one of these is important for eyesight and immune function? Yeah. Yeah. How about number 2? Which one is an antioxidant that is important for helping to make red blood cells? Yeah. It is vitamin E, yeah. So next one, which one is important for bone health and immune function? Yep, vitamin D. So now there's only one left. Which one helps with blood clotting? Anybody? I heard it. Yeah, vitamin K. Like, why did they skip all the letters in between? Oh, it's in like dark green leafy vegetables like broccoli and kale, and also I think in some other stuff. But that's what I always think of is broccoli and kale. Why? That's actually a really good question. I don't know. I should look that up. There's probably an interesting story behind that. Yeah. Yeah, it's probably, or it's like the person's last name started with a K. Like it was actually my ancestor. Yeah, no, but that is not true. Not on your exam. Um, right? Yeah. Exactly. Yeah, that would be cool. Um, any questions about fat soluble vitamins? Alrighty. So that is all we got for lipid classification. So now we'll move on to the bilayers and membrane proteins. And now, of course, I can't click on this slide. There we go. All right, so switching gears a little bit, talking more about what goes into lipid bilayers and that kind of thing. So everybody can take a couple of minutes here to sort of switch their brain over. We're going to start thinking about micelles and bilayers, what they have in common, what's different about them. So take 2 or 3 minutes and go through and answer these questions, and then we'll review them together. Yeah. Alcohol derivatives. Yeah, but used to be an alcohol and then combined it with phosphate. That was part of the phosphate.
That was
Yeah. Okay, yeah, I forgot that it's even labeled that way because I don't find that intuitive. Yesterday. Lysophospholipase.
So like when you said
Yeah.
that's what I thought of.
So I would say most antibiotics I'm familiar with don't have that type of structure. Most antibiotics generally, some of them do disrupt cell membranes, but the antibiotics that are functioning against bacteria usually do more to interrupt the cell wall. So there's a number of different classes. So like one major class that you probably heard of are like in the penicillin, like the beta lactams, right? Those all interrupt with helping the bacteria build the cell wall, so they can't divide essentially. But there are also other antibiotics that focus on other functions important for life, such as protein synthesis, or various other things like replication of DNA. But targeting that stuff is trickier because we as humans also do those things. Targeting cell wall is nice because we don't have cell walls, so it's very easy just to be like, here, take a drug that messes with your cell wall. It won't mess with my cells. I don't have cell walls. So yeah, that's the thing with the drugs. Like you want to kill the bad things, but keep the good stuff. Not just like Lysol yourself. Yeah, because cancers are easy. Yeah, so exactly. If you want to target cell replication, you want to target only the tumor cells that are replicating probably. Yep. They're more efficient, yeah, for like storing energy, I would say. Yeah. Right. I mean, that's the idea is like you can store fewer of them as energy in your lipid droplets because they take up more space, essentially. The unsaturated, right? Yeah, the saturated ones you can really cram in there, and then you can end up having very like large, very packed lipid droplets that if you use them, they're fine, but I guess they can cause you to start building them up. They're not supposed to be clots in your arteries or start getting. Yeah, I mean it's complicated. Yeah, you need some, right? But not too many. Yeah. All right. So let's start thinking about micelles and lipid bilayers. So first of all, of these two structures, A and B, which one is a micelle? Yeah, B. Yep. So which one, therefore, is a vesicle? A. Excellent. Yeah. And so the main difference about them is essentially how many layers each one of them has, right? So one of them has a bilayer. Which one is that? The vesicle. Yeah, so A is a bilayer. You can call B a monolayer if you would like to differentiate from this one, which is a bilayer. It's nice because micelle and monolayer both start with M. One way to remember the difference. The one that we're calling a vesicle, depending on how big they are, can have some other names. So yeah, so all right, so number 4 then. Which one is a liposome? Yeah, A, yeah. So a liposome is just like basically another word for a vesicle. It's a, I don't know, you can break it apart, right? So lipo meaning like fat, it's made of all these fatty acids, and with head groups, and so meaning body. So it's just like a little body made of lipids, which is very similar to a vesicle. They have different functions. Like vesicles, you find them within your cell. They're delivering proteins and things to different areas within a cell. Liposomes don't have to be inside your cells. They can be part of like a drug delivery mechanism. Some drugs are packed inside liposomes because of various reasons that we will go into as we continue to answer the rest of these questions. So before I give away more answers, let's do number 5. So which one contains lipids that have a rectangular van der Waals envelope? Yeah. A, yeah. So this is rectangular. I would also argue that it's more of like a cylinder because it's in 3D, right? They're not just flat. So yeah, so that's exactly right. So to form a bilayer, if you kind of think about taking a little maybe cross section of this bilayer, right? You have polar head groups. They're essentially forming a plane, and then their fatty acid tails are kind of filling in all that space in between. So like if you imagine this vesicle or liposome growing very large, eventually it would be like a cell. And when it's the size of a cell, the edges aren't really curving so much as almost flat, right? Like if you're like standing on one area and you look out ahead of you, it just looks like a flat plane in front of you. And so you have these, so the types of lipids that would fit in here would have 2 fatty acid tails, or hydrocarbon tails, depending what exact molecule it is. But since there's 2 of them, you essentially get a rectangle if you're looking at it flat, or if you imagine it's 3D, the shape is more like a cylinder in terms of the types of interactions that the fatty acid tails are going to be having with each other. And that's different from a micelle, right? So with a micelle, you can sort of imagine these are almost, it looks like, if you're looking at this cross section, they're like little kind of pieces of pie. And in this drawing, it's actually showing each one having 2 fatty acid tails, but a lot of times micelles are actually formed where there's a polar head group that only has 1 fatty acid tail. And so that helps them to sort of all converge in the center, the way it's shown here, right? And then each one essentially has more of that triangle, or if you think of it as 3D, more of a cone shape. For the van der Waals envelope. That makes sense to everyone. kind of. Okay, so yeah. Yes, so the rectangular slash cylinder one would be the vesicle. The triangle slash cone would be the micelle. Yep. All right, so number 6 then. Which one has amphipathic lipids in it? Yes, it is both of them. So you can see from the way they're drawn, all of them have a part that's facing the outside environment that's drawn with these little spheres. That is the hydrophilic part. It likes aqueous things, like most environments that you see biologically. And then the fatty acid tails, those are the hydrophobic part. So in the vesicle that we're looking at, you can easily see the bilayer that has hydrophilic outside, hydrophobic tails in the middle, and then hydrophobic head groups on the other side, kind of lining this middle area here, which would be, if this was a cell, that would be like the cytoplasm, right? So, all right. So that brings us to number 7. Which one of these can encapsulate a hydrophobic molecule? B, yeah. So it's the micelle. So if you think about the very center of the micelle, that's hydrophobic. So other types of molecules that are hydrophobic will like to hang out there in the middle. If you think about the center of our vesicle, that I just pointed out before, this center is lined with all these polar hydrophilic head groups. So this is going to be better at encapsulating hydrophilic things in the center. Any questions about that? Cool. Either everyone is overwhelmed or we're doing a great job. So let's keep going, unless anyone has any questions here. All right. So let's now talk about motion within a lipid bilayer membrane. So there are 2 types of motion here. So we have choice A, which is showing, you can see it pretty well in here, right? There's this orange piece here that's flipping to the other side. And in part B, we're showing this orange thing is sliding over towards the right. So, yeah, so first of all, of these 2 types of motion, which one of them is transverse diffusion? Yeah, A, yeah. So transverse diffusion is when the lipid moves from one leaflet of this bilayer membrane to the other leaflet. So now you know which this one is transverse. So what is the other type of movement called that we see in choice B? Yeah, lateral movement. Yeah, so movement from side to side. This is lateral. So you're staying on the same side, you're just moving to the left or the right, or forward or backwards, because it's in 3D, right? So now we know the two types of movement. Which one of them is more likely to occur in a bilayer? Which one is more easy to do? Yeah. Which one is easier? Which one do you think is more work? Yeah. Lateral. Yeah. So for lateral movement, your lipid is moving from side to side. All the polar heads stay next to all the other polar heads. All the tails stay next to all the tails. We're not trying to interrupt anything that's hydrophilic or hydrophobic. With this transverse movement, on the other hand, you start with this polar head group here. It has to pass through this really hydrophobic, greasy layer of tails before it can get to the other side, right? That's a lot of work, because the polar head group does not want to be in the middle. So that it can happen spontaneously, but like super slowly. It is usually catalyzed by an enzyme. Those enzymes are your flip bases, your flop bases, that kind of thing. Any questions about that? Okay. So let's go on to membrane proteins. So here is a description. I'll put up both of them, or all three of them, just so you can get a comparison of the difference. So there's 3 different types of proteins here. Is each one a peripheral, an integral, or a lipid-linked protein? So based on these descriptions, try to decipher or figure out which one is which. And I'll give everybody a couple minutes to think about that. All right. Looks like most people are kind of done. So let's start talking about these 3 types of proteins. So number 1, our G protein coupled receptor. Which type of protein is it? Anybody? You have a 1 out of 3 chance of being right. Yeah. Yeah, it's an integral membrane protein. So what clues in that little description help you figure out that it's an integral membrane protein? Yeah, so a really big clue here is it's a transmembrane protein, meaning it goes all the way through the membrane, right? Trans across, membrane, membrane. It's a big clue that it's integral. Because if we recall our other 2 types of proteins, right, our peripheral proteins do not extend into the membrane, they're just right next to it. And our lipid link proteins are attached to the membrane by a lipid. They are also not transmembrane proteins, right? Yeah, that is your big clue right there. So what about number 2? What kind of protein is a peptide hormone? Yeah, it's a peripheral protein. Yeah, and how do you know that? Exactly, yeah. So it's going to bind to the extracellular domain of another protein. So it is not itself sticking into the membrane. It's sticking to another protein that is stuck inside the membrane. So that's a very good hint that it is peripheral. So that brings us to our last one, our heterotrimeric G protein. What kind of protein is that? Yeah, it is lipid linked. And how did you know that? Exactly. Yeah, so a fatty acid and an isoprenoid are both different types of hydrocarbon linkages that we see that help attach our lipid linked proteins, help tether them to the cell membrane. So the tail of the fatty acid or the isoprenoid tail just physically stick themselves right into the membrane and interact with the fatty acid tails that are the inside components of the bilayer, and it helps. Basically, it's a little leash where the G protein is hanging off from into the middle of the cell. And so the different types of isoprenoids that you'll sometimes see, which I think I mentioned the names of, are farnesyl and geranyl, which are both derived from isoprene. And the types of fatty acids we'll see are usually myristate and palmitate, which I don't expect you to know, but if it's interesting trivia, right? Let me write that a little neater in case you want to write that down. Yeah, so those are the main types of fatty acid tethers you'll see for various types of proteins that use them. All right. Any other questions about how membrane proteins interact with cell membranes? Yeah. Yeah, so those are different types of fatty acids. So like I'm going to get this wrong. Myristate has 14 carbons and palmitate has 16 carbons. I'm 95% sure, or it's the other way around, but I'm pretty sure it's definitely 14 and 16. Pretty sure myristate's the 14 and palmitate's the 16. And your 18 carbon one is stearic acid that we've talked about a few times in the lecture. Stearic acid is also a fatty acid. It's not usually found in tethers, but it's frequently found in like triglycerides and in sometimes it's one of the fatty acids in your cell membrane. So yeah, we'll see stearic acid a lot. It's the 18 carbon saturated fatty acid. All right. Any more questions about that? Cool. So that will bring us to the next section, which is about membrane structure and assembly. So let's start thinking about the fluid mosaic model. So we were just talking about types of movement in membranes. So everybody take a couple minutes and think about what components of a membrane are mobile and can undergo lateral diffusion. All right, so everybody should know at least one component of a membrane that undergoes lateral diffusion, because we were just talking about it a few minutes ago. So does anybody want to volunteer what any of those components are? Yeah, so proteins are definitely one of them, especially when they are attached. Is there any time when proteins are proteins always mobile, or can they sometimes not undergo lateral diffusion? Yeah. Yeah, not always. Yeah. Do you know why they are not always? It's okay. Yeah. Anybody else? Yeah, essentially. So proteins, so usually when we're talking about movement, we'll talk about movement being constrained. I did not spell that right. Constrained. Okay. And so something that proteins can be constrained by is the membrane skeleton, which I'm not sure if I mentioned in the lecture. So at this point, it's probably good to point out that cells are not just like bags of water surrounded by membranes, right? Cells themselves have a lot of structure to them. They have to maintain their shape, and they have to also provide essentially like a scaffold on the inside to allow all the little components inside to move and talk to each other and all that good stuff. And so cells have a lot of structural components, like you probably heard of cytoskeletons, and in particular, some cells or some compartments of cells also have a membrane skeleton, which is basically just like a rigid part underneath the membrane where some things can be attached to or they might not be attached to. So if there's a membrane protein that is attached underneath to the membrane skeleton, that protein is not able to move laterally across the surface of the cell because it's stuck inside this little scaffolding underneath, essentially. But not all membrane proteins are constrained. Only some are and some aren't. So that brings me to the rest of this question. So what other components of a membrane are mobile besides proteins? What else is in membranes? What are membranes made of? And is it mobile? Yeah, lipids. So we were just talking about a few questions ago how lipids can freely laterally diffuse for the most part, unless they can also be constrained, actually, in different ways. And this type of lateral diffusion is much easier to do than that transverse diffusion where they flip across to the other side of the membrane, right? So that brings us to the second part of this question, which is, what does FRAP stand for and how is it used to observe lipid and protein diffusion? So everybody take a few minutes, think about what FRAP is. All right, so in the interest of time, I'm actually we'll pick up here on Thursday because there's only a couple minutes left, and I want to talk for more than a couple minutes about FRAP. So we'll save that for Thursday. I think you all are also information overload a little bit, so let's let everything sink into your brains. I'll see you on Thursday. Don't forget your next quiz is due Thursday night. For those of you in the lab, your LDH lab report is due on Wednesday. And if anyone has to do their paraphrasing revision, that's due tonight. So enjoy, and I will see you all on Thursday.